Hello friends, this video on NEET Biotechnology is brought to you by examfear.com. No more fear from exam. Question number 15. Plasmid is fragment of DNA which acts as vector, a fragment which joins two genes, mRNA which acts as carrier, autotrophic fragment. Well, plasmid is a small fragment of DNA which are like vehicles for gene transfer. So they facilitate transfer of genes. So they are vectors. So it's the first one. Question number 16. Two bacteria found to be very useful in genetic engineering experiments are Nitrosomonas and Klebsiella, Escherichia and Agrobacterium, Nitrobacter and Azotobacter, Rhizobium and Diplococcus. Now, when you talk about genetic engineering, for genetic engineering in animals, for example, insulin production, so the most commonly used bacteria is E. coli, that is Escherichia. Whereas when it comes to genetic engineering in plants, it is mostly Agrobacterium. So in fact, Agrobacterium is a pathogen of many dicot plants. That is, it causes diseases in many dicot plants. It transfers the tDNA to convert normal plant cells into tumors. So basically, it is the cause of tumors in plants. But at the same time, the plasmid of Agrobacterium has been modified into cloning vector that is not pathogenic to plants. So that ways, Agrobacterium is widely used in the genetic engineering experiments. So the correct option is B. Question number 17. The first human hormone produced by recombinant DNA technology is insulin, estrogen, thyroxine or progesterone. So obviously it is insulin. In fact, we have learned in detail about the genetically engineered insulin, and this hormone controls the blood sugar level. Question number 18. Consumption of which one of the following foods can prevent the kind of blindness associated with vitamin E deficiency? Flavor saver tomato, canola, golden rice or BT brinjal? Now, so we basically need to tell which of these options is rich in vitamin A, because it has to prevent the kind of blindness that happens due to vitamin A deficiency. That means the person should take more and more vitamin A rich food. So out of the options which are given here, golden rice is rich in vitamin E. Now normally the rice which is normally available, that doesn't contain vitamin A, but genetically engineered golden rice. So golden rice is Oryza sativa. So this is rich in beta carotene, which is synthesized by genetic engineering, and this beta carotene is a precursor of vitamin A. So golden rice is rich in vitamin A. Now what is the benefit of making this golden rice? Because as rice is a staple food, so if rice is made to contain vitamin A, what would happen? The children would eat rice, so as a result they would get a lot of vitamin E inside their body, and this would prevent these kind of blindness diseases which are generally associated with deficiency of vitamin A. Question number 19. Maximum number of existing transgenic animals is of fish, mice, cow or pig. So maximum number of transgenic animals are that of mice. So transgenic animals, we all know that here the DNA is manipulated to possess and express a foreign gene. So that's transgenic. Question number 20. Genetically modified brinjal in India has been developed for insect resistance, enhancing shelf life, enhancing mineral content, drought resistance. So here the correct option is A, that is insect resistance. Now this genetically modified brinjal is basically BT brinjal. And what is BT? BT stands for Bacillus thuringiensis. So this BT produces an insecticidal cry protein. So this cry protein is insecticidal in nature. That means it is capable of killing insects. So when you have this variety of brinjal, the variety itself is resistant to insects. So that's the main advantage of the genetically modified brinjal. Question number 21. An improved variety of transgenic basmati rice does not require chemical fertilizers and growth hormones, not really, gives high yield and is rich in vitamin A. Yes, this is absolutely correct. So this is what we were talking about, that this transgenic variety of basmati rice or the golden rice, which it is more popularly known as golden rice, the biological name is Oryza sativa. So it is genetically modified to biosynthesize beta carotene, which is a precursor of vitamin A. In fact, Professor Potricus and Beer produced genetically engineered rice by introducing 3 genes associated with the synthesis of carotene. So that's how golden rice was synthesized for the first time. Question number 22. A transgenic food crop which may help in solving the problem of night blindness in developing countries is BT soybean, golden rice, flavor saver tomatoes, Starlink maize. Well, again, night blindness is caused due to deficiency of vitamin A. So out of these, what is rich in vitamin A? Golden rice is rich in vitamin A. So golden rice can solve the problem of night blindness. Now, if you talk about the BT soybean, anything which has BT, that is Bacillus thuringiensis, BT is insecticidal in nature. So BT soybean would be insect resistant. Now, if you talk about flavor saver tomatoes, so these are genetically modified tomatoes which remain fresh for a longer period of time. In fact, they have a superior taste as well. So how does that happen? So why is it that they remain fresh for a longer period of time? Because the gene producing polygalacturonase is inactivated, and this polygalacturonase causes over ripening or ripening of the tomatoes. Now, since this gene is inactivated, therefore the over ripening is prevented and therefore they remain fresh for a longer period of time. Question number 23. Production of a human protein in bacteria by genetic engineering is possible because bacterial cell can carry out the RNA splicing reactions, the human chromosome can replicate in bacterial cell, the mechanism of gene regulation is identical in humans and bacteria, the genetic code is universal. Well, here the correct option would be D, that's genetic code is universal. Now, what is genetic code? It is a common language for almost all the organisms to translate DNA or RNA sequences into amino acid sequences. So that is the purpose of having a universal. That's the purpose of having a universal genetic code, that it helps to translate the DNA or RNA sequences into amino acid sequences. So the 3-letter codon represent the same amino acid for every organism on earth. For example, you have the 3-letter sequences like GAA or AUG or GUA. So each of these codons, so these 3 letters together is called codon, and each codon represent a particular amino acid. And for every or most of the organisms on this earth, a codon represent the same amino acid. So that shows that the genetic code is universal, and therefore it is possible to produce human protein in bacteria by genetic engineering. Question number 24. Producing a giant mouse in the laboratory was possible through gene mutation, gene manipulation, gene synthesis, or gene duplication. So of course by gene manipulation, because how do we produce a giant mouse? By gene transfer to fertilized eggs and stem cells. So that's how we are going to manipulate the kind of organism we want. So we basically want a mouse, but we want a giant sized mouse. So the gene which are responsible for the size of the mouse, that needs to be manipulated. So with gene manipulation we can get a giant mouse in the laboratory. Question number 25. The transgenic animals are those which have foreign DNA in some of its cells, foreign DNA in all of its cells, foreign RNA in all of its cells, RNA and DNA both in the cells. Now, transgenic animals means that they have foreign DNA in all of its cells. So, how do we produce transgenic animals? So, production of transgenic animals include identifying and separating a desired gene, which will act as the foreign gene. Now select the proper vector which will carry that gene or which will help in transfer of that gene. So basically the first step would be to select a desired gene. So this gene will later become the foreign gene. Second step would be to select a proper vector which will carry the gene or which will facilitate gene transfer. And then the third step would be combine the desired gene with the vector. So basically 1 plus 2, that means this desired gene, combine it with this vector. Okay, now the fourth step would be to introduce this vector in an individual. So introduce the vector into that organism where we want to put that desired gene. And finally the last step would be expression of the foreign gene. So now in that individual, this vector and this gene is like a foreign gene, right? So now the foreign gene will express itself in that individual. That means you will be able to see some different traits in that individual or that organism because of the presence of this foreign gene. And then that animal is said to be a transgenic animal. Right, so these are like in simple words, this is how it is. So basically transgenic animal should have that foreign DNA, like this foreign gene has to be present in all the cells of that transgenic animal. Thank you. Please visit examfear.com for free quality education. You can learn with a simple 4-step learning process wherein you can watch video lessons, you can ask your questions, you can refer notes, and you can take a free online test. We have content for class 6 to 12 on physics, chemistry, mathematics, and biology along with practical videos. So please subscribe to our channel for daily updates. Thank you.